Hi everyone, today we are going to talk in detail about Kinocilia versus Sterocilia differences. So let's start the topic with introduction. Kinocilia and Sterocilia are specialized apical surface modifications that are found on certain epithelial cells. Both appear as hair-like projections under the light microscope, but they differ significantly in their structure, internal composition, motility, and functions. Kinocilia are motile structures that are responsible for movement of fluid and particles across epithelial surface, while sterocilia are non-motile structures that are mainly involved in absorption or sensory function. Basic nature of structure. Talking about kinocilia, kinocilia are true cilia that project from the apical surface of epithelial cells. They are capable of rhythmic beating movements that help transport substance across the epithelial surfaces. Second is sterocilia. Sterocilia are long, non-motile projection that resemble cilia but are structurally different. They are actually elongated microvilli and they do not produce active movement. Talking about second characteristic, that is internal structure and composition. First, talking about kinocilia. Kinocilia contain microtubules arranged in a characteristic 9 plus 2 pattern. This arrangement consists of 9 peripheral doublet microtubules that surround the 2 central microtubules. This microtubule arrangement forms the axoneme, which is responsible for motility of cilium. Talking about sterocilia, sterocilia do not contain microtubules. Instead, they consist of central bundle of actin microfilament. Because they are composed of actin microfilaments, they are structurally similar to microvilli rather than cilia. Talking about third characteristics, that is motility. Kinocilia are motile structure. They beat in coordinated manner, though two phases known as the effective stroke and recovery stroke. This movement produces waves that move substances across the epithelial surfaces, while sterocilia are non-motile and they do not beat or move actively. Their function is mainly related to absorption or sensory activity rather than movement. Third characteristic is length and appearance. Talking about kinocilia, they are usually shorter and more uniform in length, measuring about 5 to 10 micrometer. They appear as fine hair-like projections that are arranged in rows on the epithelial surface, while sterocilia are much longer than kinocilia and may reach a length of up to 30 micrometer. They often appear as long, irregular, tuft-like projections. Next characteristic is presence of basal body. Talking about kinocilia, each kinocilium originates from a basal body that is located in the apical cytoplasm of epithelial cells, and the basal body has a structure similar to centriole and contains 9 triplets of microtubule, that is 9 plus 0 pattern. While sterocilia do not arise from basal body, they are extension of cell membrane that are supported by actin filament that extend into the cytoplasm. Location in body. Kinocilia are mainly found in epithelial tissue where movement of substances is required, such as respiratory tract epithelium and uterine tubes, and ependymal lining of brain ventricles and sensory cells of inner ear. While sterocilia are present mostly in epidermis, vas deferens, and hair cells of inner ear and certain sensory organs. Functions of kinocilia versus sterocilia. Kinocilia primary function is movement of substances across epithelial surfaces. In the respiratory tract, they help move mucus, dust, and microorganisms towards the throat. In the uterine tube, they assist in transport of the ovum. While sterocilia function is mainly depend on their location, as in the epididymis, they increase surface area for absorption of fluid and sperm maturation. While in the inner ear, they are involved in sensory signal generation and they help convert mechanical stimuli into nerve impulses. And this was all about differences in kinocilia versus sterocilia. Thank you for watching.